Okay. Hi.
Hello.
Hello from Germany. I'm very happy to introduce, first of all, our webinar series, The Rise of the Live Cell Shipment. And today, our focus is on the logistic and the importance for organ on a chip. And because this is really a very, very important topic for us, we welcome our partner, or my speaker, Dr. Rake Holland, who is, and congratulations, I heard that this is brand new. So you are the new CEO of Tissues GmbH, located in Munich. And you were actually already in the company being the head of business development. So first of all, congratulations. I'm very happy that you are here today. And before I hand over to you, I just introduce myself. My name is Katrin Adelkofer. I'm the founder and CEO of Cellbox Solution. It's also a small company from Germany, located in Kiel and in Hamburg. And we are actually focusing on technology which enables cells to travel safe and sound from A to B, not frozen, but alive. And that is not only cells, but it's also organoids, 3D structures, fragile cell complexes. And we see the upcoming need that the transportation is not only done at room temperature or frozen because you lose quality and quantity. And with our technology, our Cellbox, we believe that we can actually close the gap from one company to another, from on a campus by flight or by ground, the gap to transport. And this is what we actually want to understand what it's all about, and what are actually the challenges in this regard of logistics. And since that, we would like to establish a cell traveler community. And if someone is bringing a friend to the next webinar, which I think will be in the beginning of November, they will get, if they bring a friend, one of these little cell boxes. They are not the normal size. These are just little tiny boxes. If you have 10 of these, you can actually get one of the Cellbox bags. So actually, I hope this is interesting enough next time to bring one of your colleagues or someone you believe who might be also interested in our webinar series. Before I start questioning you, I would be very happy, like if you could introduce yourself, your person, and then also, of course, giving us a short introduction about Tissues GmbH. Okay, so it's yours now.
Yeah. So thank you very much for the nice introduction. So yeah, my name, as mentioned, is Rake Holland. I'm currently the CEO of Tissues. I'm a biotechnologist by training. Studied here in Berlin, where I then specialized in medical biotechnology. Did my PhD then in tissue engineering, where I tried to grow hair in vitro in the cell culture laboratory. Not as successful as some may think. It was in a way a success. It's not a transplantation method yet, but we use it sometimes to test in vitro for hair growth promoting substances. Yeah, and that was also the stepping stone to move into this multi-organ chip, organ-on-a-chip project, which we started in 2010, again at the Technical University of Berlin, which was funded by a BMBF Go Bio grant here in Germany, where we had a couple of years to develop the technology platform. And then since around 2014-15, brought the first products to the market. And we are now active with a lot of customers and partners in the pharmaceutical industry, in the cosmetics industry, chemical industry, wherever there is a need to test or to find new methods to test substances in a more predictive manner compared to the currently available systems. That's where we try to step in with our multi-organ chip platform and help our partners to achieve their research and development goals. Now, we do have a couple of different products on the market. Our focus is on multi-organ chips. So even though each of the chips which we have, you can also use as single organ chips, we are dedicated to building multi-organ chips in order to understand the systemic interaction of the organs, much like they are arranged in the human body, interconnected by the vasculature system. That's how we also want to mimic it in our chip systems. We do have chips available where you can combine 2, 3, and 4 different organ models that are now working on a chip system, which can combine up to 11 different organs in one chip.
Very good. So when you look at your application, what is the application where it is needed most in the moment? In which field of the market?
So I mean, when we started, the first clients which we actually had was in the cosmetics industry, because you have a ban on animal testing since 2013 in the European Union. So there definitely is a need for new methods on how to test new cosmetic ingredients. On the other hand, you also, of course, have the pharmaceutical industry, where you probably also have seen that these slides always come up in one presentation or the other about the failure rate, the attrition rate the pharmaceutical R&D has, especially in the clinical trials, where you say approximately 8 out of 10 compounds which enter the clinical trials, they will fail. So you end up with 1 or 2 compounds on the market, and this is of course a huge attrition and failure rate associated with high costs. So this is also a market which is highly highly interesting for us. And these attrition rates are now for I would call it the historically grown small molecule entities. And now we are moving to new therapeutic models with cell therapies, with immune therapies, which are specifically tailored to the human situation, where it's getting even more and more difficult to detect something in an animal model because it's specific to human. So there's even higher need to find new alternative methods to generate data which is meaningful and predictive compared to what you will see in the first clinical trials when you do the first tests in humans.
If I come to that in a moment, but maybe a very quick question regarding, so years ago or still, we have the like the normal cell culture, like system. It's like a one layer, and you are working not only in a 3D model, but you also align organs. So what do you think is actually the advantage that you say that you form little organoids, you put them into a chip. So what is the advantage of a liver cell layer or an organoid structure? Is there a difference in how they are, how they interact?
So, the easiest comparison is always that you can say there's actually no tissue or organ in the human body which is two-dimensional, so consisting of just one cell layer. We always have three-dimensional organs, and that's why, of course, 3D models are physiologically more relevant compared to 2D models. However, we also do not see this as a kind of black and white thing, because 2D models also have advantages over 3D models if you think, for instance, about throughput. So organ on a chip systems or also more complex 3D models it will be difficult with them to achieve the same throughput as you can with just a monolayer of cells in a kind of high throughput system. So they each have their place. You can use the 2D systems to, detect or select substances, promising candidates, when you still have, 10,000 or more candidates. But the more you move closer to the decision-making point where you need to decide whether to move it into the clinic or not, Yeah, that's where you need systems of a higher complexity, which are capable of producing these more predictive data, which are more relevant to the human situation. And that's where these 3D models come in, which can either be used outside, definitely outside of an organ-on-a-chip system, or of course, where the organ-on-a-chip systems come in with their advantage of giving mechanical cues to the cells, which are in a lot of different organs, are very important factors which bring in the flow. And the flow, which I mean, we have learned this from the bioreactor, from the standard bioreactor systems. Perfusion systems give better nutrient and oxygen supply to the cells compared to standard static cell culture systems. So you can keep the cells viable over longer periods of time compared to static cell culture models. And then in our case, like I mentioned, you have the option of connecting these tissue and organ models with a kind of a vasculature system, much like they are connected in the human body. So you also get some systemic readout. You don't just get to know what the liver is doing. Is the compound liver toxic, or what metabolites is the liver producing or generating from that compound? You can also see an immediate reaction, for instance, if that metabolite which the liver is producing has a neurotoxic effect, is not excreted efficiently by the kidney, and so on. So these are then the huge advantages where you usually would need an animal model, and we all know that we are not a 70-kilogram rat or mice. We, in a lot of ways, work differently and behave differently than the animal models or the animals which we are using for these trials.
So one more question regarding the structure. So you're saying 3D structure versus 2D, but then you also have like organs which are consisting of not only one cell line. So you, for example, have maybe multiple cells or different cells within one little organoid on your chip. Can you comment on that? Because I think this is something which is very hard to achieve in a cell culture.
But yeah, it's very important actually that, and you're absolutely right. You should take a look at the at the physiology of these different organs in vivo, and decide which cell types you would like or you should include. This can be a certain aspect with regards to the context of use. Yeah, if you just need, for instance, in the liver, the hepatocytes to answer your research question, then you don't need to make it more complex. But in a lot of cases, for instance, you need the stellate cells, you need Kupfer cells, you need maybe sinusoidal endothelial cells, and then you need to add that. And that's also what we always try to aim for in our multi-organ chip setup, that our models consists of the different important cell types of the organs. And this is something which kind of drove us from the beginning, and we did a lot of literature research in the beginning. And interestingly enough, you actually have to go back to the publications from the 1950s and 1960s to get data and information on the ratio of the different cells in the organs. You don't find that in the newer publications anymore. It's always about, function of the cells, how certain things are working with each other, but physiology is only described in these very old publications, but then very, very correctly. And these information you can then take out, and with a certain downscaling ratio, of course, can then use also in the chip system, so that you have a defined ratio of different cell types in the models which you are creating. For instance, if you think about an organoid or a spheroid culture, you can set up that spheroid culture in a way that this represents the ratios, the in vivo relevant ratios of these cell types.
Yeah. So what I like about your company is that you're actually avoiding, or at least at one point, like the animal models. You don't need to make so much animal models or testing on animals if your system is working well, because you can already predict much earlier, because you can do the metabolites, you can analyze the metabolites, you can see if they are toxic or not. So I think in the preclinical state, if you have such a like kind of little organism with all the different organoids aligned, you can actually avoid working with so many animals. This is what I learned about your chips. I have one question. So if you have all these chips nicely made in your company, so what about the logistic? And this is what we are talking about. It's like you have them there, but you need to maybe, and this could be one model, is that someone else does need the organ on a chip, or these chips need them somewhere else. How do you do that? I mean,
So,
yeah.
Yeah, so at the moment, it's definitely in most cases the situation is that we ship the cells, the chips without cells. So the partners, the customers get the chips and basically an application note telling them how to prepare the chips, how to prepare the cells, how to bring the cells in, and so on. Naturally, of course, there are lots of requests on can't you just ship the chips already with cells in them, so we can just unpack them, plug them into the pumping devices, and then start our experiments, which of course would save a lot of time. Anybody who has already ordered maybe not only cells, but maybe even tissue and organ models like skin models or liver spheroid models or whatever, may already know that, especially if you think about international transports, customs can be a big hurdle there. Yeah, for whatever reasons, then these shipments are held up at customs. The ice is melting. All of a sudden, they are cultivated in room temperature. Maybe the media is not perfect anymore because the cells are still consuming the media. Oxygen is may change and everything, and this can lead definitely to damages to the cells once you eventually arrive at your site, that you cannot use these cells anymore. And now we are talking here about not only cells or one tissue model, but in our case up to 4, and maybe in the future up to 11 different organ models, which are simultaneously on one chip. Each and every of these models maybe with a little bit of different requirements and so on when it comes to shipment. So the ideal solution, of course, would be to give them the same conditions, the same environment as they have in the laboratory. And that's why we are currently taking a good look at options on how we can achieve this. And Cellbox, for instance, could be one of these solutions where we say, okay, maybe even ideally with a pumping mechanism integrated, so we can also keep the pump running, that we can ship these chips then with a solution which is capable of transporting and bringing the chips together with the cells to the clients, so they really just have to plug them in and can do their experiments.
Because I remember when I was young working in drug discovery, it was always about the pressure molecules, which you definitely don't want to give away. You want to keep it in-house, and you better do your screening in-house. So that's why all big pharma industries started to screen their own libraries, because this was very hard to, transfer libraries to someone else. And exactly in your case, ideally, you can prepare these chips and just send it so they can do their own experiment without doing a whole cell biology PhD, because this is something which you are specialized in. And I can only vision how difficult it is to set up such a chip. And looking in the personalized medicine, so it's very individual, so that basically you can create chips on a personal level. Is that something you are already looking into? Because this is a very upcoming field.
Yeah, so yeah, that's definitely something. I mean, we have published, it was last year, the first paper with the chip4 system, where we combined 4 different organ models: an intestine, a liver, a kidney, and a neuronal model from the same donor, all iPS derived models, on a single chip, and basically created an autologous donor specific chip where we could in theory, then detect or define the reactions of that specific donor to a certain treatment. And if we would have disease models, of course, could also then detect how the donor is reacting to the treatment, if we can cure the disease or prevent the disease from spreading. So that's definitely something which we are looking at. And organ-on-a-chip systems and personalized healthcare, personalized medicine for sure is a big topic, because as soon as you are able, especially to bring these multi-organ chips to a personalized level, you could even imagine not only reducing animal tests, but also substituting clinical trials, because you are then able to already in the preclinic to select your donor, your donors from the IPS bank based on gender, on genotypic background, on age. So you maybe have a 53-year-old male smoker and a 25-year-old female non-smoker, and set your surroundings, and then test on these chips these different donors, and basically can get data which you would only be getting in the clinical trials when you select your participants in these clinical trials based on certain criteria. And this can then be really leading to a paradigm shift on how we are testing drugs, especially with regards to the time and the money which is necessary to bring a drug to the market.
So I imagine, like, for example, if you go in phase 1, instead of giving the drug to the patient, which you would be allowed, you would give it on a chip. And the chip would consist of personalized organoids from the individual you normally would treat. So that the first step could be even one more safety step. So looking at the individual before you go on patients, that would be one possibility. So how fast can you? So if I would ask for, can you prepare for me a chip, not having anything? So how long would that take? And all over.
I mean, we are pretty fast with producing the chips. I think the time limiting point is still producing the IPS cell line from you, and then a little bit more time to differentiate then your different tissues out of these IPS cell line. So there definitely needs to be more progress in the way on how we generate these IPS cell lines, because this can take a couple of months. And if you are sick, you don't want to wait a couple of months before you start finding about different treatments. However, there's also some kind of intermediary solution where you basically say you have a certain donor bank already. We could check for which is maybe someone who is close to your situation. Use that bank to build kind of the standard organs like liver, intestine, kidney, maybe in that case, and just integrate, for instance, if you have a tumor, the tumor model from you in the chip system, in order to get already some understanding on how the treatment may work. This is kind of half personalized, but at the moment it's the faster solution compared to a fully autologous chip, where, like I said, it still needs some progress and advancement in how we generate these IPS cells.
So how many chips can you build a day? I mean, not this individualized, but if someone would say, I need a 4-way organ and ready to be screened or ready to be analyzed, how many can you do in a week?
In a week, I think we can up to 100, 200 chips, even more. It depends on the manufacturing and how many people you put into production of the chips. So it's definitely scalable, and we believe that we can at least with the capacities which we currently have in-house, we can produce internally up to 15,000 chips per year. So that would be the maximum before we need to expand.
Yeah, no, I'm I'm wondering, especially when you now with possibility using Cellbox or something where you can ship them alive. This is a totally new business model. I mean, before that, you were able to analyze. So people were sending you the compounds, the chemical you need to analyze, and you did a service. Now you can actually expand your business and scale it in a way that you can send the chips all over the world. If, of course, the system is working and you can transport them in this ambient situation. So that actually gives companies like you also, a new business model or something which you could scale up in that way. I actually wonder, did you ever, did you see that you can actually even go that far that these organ on a chip can replace part of the clinical trials? I mean, could you see that, for example, if pharma industry, would use these kind of chips, could we actually come to the place that we can make clinical trials cheaper?
Cheaper, yes. That's what we believe, because what we believe is that we definitely can substitute the clinical trials, and this is then all a matter about regulatory acceptance of this data. If this data then is regulatorily accepted, then definitely the clinical trials could be cheaper. If I wouldn't go as far as saying you could completely replace it, because I mean, what we can build is, even in the future with this chip which can hold 11 organs, it's still a mini organism, a minimal organism compared to the complete human. So that's what it's not in a way one-to-one comparable. So definitely there still need to be clinical trials, but the chip systems definitely have the capacity and the power to substitute and to add to these clinical trials.
Yeah, no, I'm very curious to see how tissues is going to evolve. I mean, you already did already exciting steps. I watch you since a long time, and we are also very happy if we can help you. So Cellbox is when you said, for example, how can we make them even more efficient, working with pumps so that while they are transporting, they still get enough nutrition. I mean, we are all in the field where we have to find solutions of while traveling that the cells are the most happiest ones, so that at the end point that they will actually be there having the same results as if you would do it in your in-house in your laboratories. We are almost finished, and I have to announce, first of all, you get, because you became CEO, we sent you one of these bags. Yeah.
Thank you so much.
It's not a box, and it's not this one, but it's a bag. Thank you so much for being here today with us. We wish you all the best for your new job, your new role. I hope that we see each other in person at one point. And I just should say that we should all stay tuned, because we will have a webinar this year. I think it will be in November. And yeah, and I hope to see you, like, as soon as possible at one point. And good luck to your whole team. And thank you very much. And thank you for all of you of being our guests today. And good luck for everybody, and stay healthy. Bye. Cheers.
Thank you. Bye-bye.
Bye-bye. Bye-bye.